Hello my dear friends, welcome to LivePrep. Now we are starting with a new chapter, that is the cell cycle and cell division. The chapter is very small, but conceptually it is very strong chapter. So here generally what many students do is they will try to by heart the information related to each phase. But my suggestion is don't try to by heart. There is a sequential phases are present. Sequentially there are some phases. So try to understand how exactly the changes are taking place in the each phase. So if you are keep observing on this, and if you are practicing, and if you are start imagining about these things, then this will be the most easiest chapter. Anyhow, the weightage is also very high. So you need to understand everything by a sequential process. So that's why my suggestion is don't try to by heart the content. Try to understand how exactly the changes are taking place one after the other in different phases. That's it. So that it will become very easy. But anyhow, in this lecture, so we are studying some general introductory information. So original information, so slowly we will start from the next lecture onwards. Clear? So now we are going with this particular one. So now first we need to talk about the cell theory. Who proposed the cell theory? Schleider and Schwann proposed the cell theory. Already in the cell, the unit of life chapter, we have studied this. And originally when they proposed only one point. What is that? The organisms, all the living organisms are composed of cells and their products. That's it. That's it. And now then what we have studied, so we have studied about another scientist, and his name is Rudolf Virchow. And what he has proposed? He proposed that new cells are formed from pre-existing cells by cell division. Cell division, that means who first proposed about the cell division concept? It is about Rudolf Virchow. So please understand this. Rudolf Virchow proposed that cells, suppose this is a parent cell, it will undergo division so that it is forming the daughter cells. These are called as daughter cells. Please understand the words also. So the cell that is undergoing the division is called as the parental cell, and the newly formed cells after completion of the division are called as the daughter cells, or you can also call these as progeny cells. It is the progeny cells, yes or no? So after the division, whatever the cells that are produced, they are called as progeny cells. Now this is one round of division. Suppose these cells are further undergoing division. Now what happens? From this one again, two cells are produced. From this again, two cells are produced. Now in this case, for these cells, these are the parental cells, and now these are the new progeny cells. So in this way, generation after generation. So now this is the parent cell. So the cells that are produced will be the upon division, they are called the first generation cells, and when they are undergoing the division, they will form the second generation cells. So in this way what happens, these cells will keep on dividing and dividing, so that from the initial single cell, so if the division is keep on happening, even the millions of billions of cells also can be produced. So this is what the basic logic about. So if you are generally we are studying with respect to the human, please remember, in majority of the cases I will take the example of human, so that you can easily understand the concept. Okay, so if you are talking about the human, in the case of human, what happens? Zygote. Already at the end of the cell chapter I told you about this one, right? So sperm cell, and then the ovum, they are undergoing the fusion, so that they are forming single cell called zygote is a single cell. And this diploid zygote, this is the ploidy is diploid because sperm is haploid, ovum is haploid. Upon the fertilization of these two, what happens? Diploid zygote is produced. This is a single cell, and now this single cell is dividing for several times, so that ultimately it is forming a complete individual, and the individual is having billions of cells. Now you just imagine how these many cells are produced from this single initial cell. This is all by the division of the cell. So this is undergoing division and forming two cells. Again they are undergoing division, again they are undergoing division for several times, so that now you can see billions of cells in single human individual. So this is in this way, not just for human. Now you just see any of the organisms. In the case of multicellular organisms, if you see, majority of all these things, so they are starting from single cell. So single cell, they are starting their life from the single cell. It is keep on dividing and dividing, so that ultimately it is forming a multicellular organism. So in this way, any multicellular organism you just see, this is what the case is. So that is the reason why this cell division is very, very important in the formation of individual. So in this way, in the NCERT, they are trying to give some basic introduction. So first we will go through some basic introduction, then I will slowly explain each and every point. Clear? So first let us go through some basic information given in the NCERT. So this is what the NCERT example they have given. Don't worry about the cycle and the division differences. Slowly I will tell you all the points. Here they have given some introduction. Are you aware that all organisms, even the largest, start their life from a single cell? What is the largest organism here? It is the blue whale. Please understand. So largest organism is the blue whale in general. Suppose they are asking separately for the land and water. In the water, if they are asking again blue whale, on the land it will be the elephant. Okay. So these are the largest organisms. But anyhow, overall if you consider, again it will be the blue whale. Okay. Even the largest starts their life from a single cell. What their single cell is called as zygote. They are starting their life from single cell called as the zygote. How this single cell is developing into these many cells? It is by the division. That's what they are saying. You may wonder how a single cell then goes on to form such a large organism. How the single cell is able to form this much of large organism? Because of the cell division. Next one, growth and reproduction are the characteristics of the cells. So cells will grow in size. Once they are growing in size, then they are undergoing the division so that they are forming next generation cells. That forming the next generation cells is nothing but reproduction for the cell. For the cell, forming next generation cell is called as reproduction of the cell. So in this way, growth and reproduction are the characteristic features of the cells. Indeed, of living organisms. So they are the characteristic features of the cells and also the characteristic features of the organisms also. Organisms are also going under growth and organisms are also form next generation organisms, that is called reproduction. So cells also showing growth and reproduction. Organisms are also showing growth and reproduction. Anyhow, regarding this particular content, so you will get some basic introduction in the living world chapter itself, right? Now let us continue further. All cells reproduce by dividing into two. All the cells reproduce by dividing into two. Now this will be the point. So here I will give some basic information. So please understand carefully. Here we are talking about cell cycle and cell division. Cell division. This is what the heading is. Please understand, here we are talking some technical details. And just know what is the sentence we studied. Cell will undergo divide into two. Cell will divide into two. Why only into two? So now we will see about what is the information we know. We know about two types of cell divisions. What are those? One is called mitosis and other one is called meiosis. In this way, you know about two types of cell divisions. In the case of mitosis, what happens? Single cell will divide into two cells. And in the case of meiosis, what happens? Single cell will ultimately divide into four cells. This is what, right? So please understand, in the mitosis, what happens? Single cell will form two daughter cells. But in the case of meiosis, single cell will produce four daughter cells. And here you need to understand that why in the NCERT they are talking about one is dividing into two. Please understand this concept carefully. I will tell you each and every interlinked information. All the cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. So every time when they are dividing, one parent cell is dividing into two daughter cells. Why they are saying only about this one? Yes or no? This is what they are saying, no? One parent cell will divide into two daughter cells. Suppose this is the parent cell, again this will form only two cells. In this way, they are talking only with respect to mitosis. Why they are not talking about the meiosis? This is the first point that you should think on it. The logical reason behind this one is, here they are talking about the zygote. Zygote is developing into complete organism. When zygote is undergoing the division, what type of divisions it is undergoing to form this complete individual here? So in the majority of it, there are some rare exceptions where zygote can undergo meiosis. That is a different case. So in the plants you will study, so that is a different case. But we are talking about general information. In the general information, what happens? Zygote is a normal diploid cell and the zygote is undergoing mitosis. mitosis, so that it is forming two daughter cells. Again these cells will act as parents, so that they again form two cells. So in this way, every time in each division what happens, from one cell two cells are produced. So in this way they are forming the complete organism. Please understand, this is the first point you need to understand. So for the formation of complete organism, what type of division is required? Mitotic divisions are required. Clear? And the next important point is, here they are saying that in the case of mitosis, what happens? Whatever is the ploidy of parent cell, the same is the ploidy for daughter cells also. Yes or no? But in the case of meiosis, what happens? Whatever is the ploidy of the parent cell, the ploidy of daughter cells is off of that. If the parent cell is 2n, so daughter cells are off of it. That means they are only haploid. If the parent cell diploid, these are haploids. That too. Here, if you are observing here they used a term called cell cycle. Cell cycle. Please understand. When you will use the term called cycle? When we will use the term called cycle? Suppose there are some sequence of events are present. And if they are repeated again and again, then only the cycle term is used. Please understand this. There are some sequence of events. If the sequence of events are repeated in the several rounds, then what happens? Then only it is called as a cycle. Let us take simple example, Krebs cycle. In the respiration, you will study about the Krebs cycle. What you will study? There are some sequence of reactions are taking place here. And now they are repeated. That's why since they are repeated, it is called as Krebs cycle. Understood or not? Can you call glycolysis as a cycle? Is it like a continuous circular pathway? No, it is not like this. It is a sequential process. That's it. So that's why for the glycolysis, we are not using the term called cycle. But for the Krebs cycle, we are using cycle because sequentially the same things will be repeated for several times. In the same way, here also we are using a term called cell cycle. In the cell cycle, what happens? There are phases called G1 phase, S phase, G2 phase, and then M phase. At the end of the M phase again, two cells are produced. I am talking for mitosis. And again what happens? This cell, it is present in G1. Again it will undergo S phase, G2 phase, M phase, and form again two cells. Same way, this is also producing another two cells. Again now these cells will undergo division. So in this way what happens? Continuously they are able to divide. That means the term cycle is used only for mitosis. Technically, if you are observing, the term cycle is used only for mitosis. Cell cycle term you should not use for meiosis because in the meiosis what happens? Suppose you are starting with the diploid one. This is in the G1 phase and then S phase, and here generally G2 is not there. So directly we are going with meiosis 1. At the end of meiosis 1, what happens? Two cells are produced and again they will undergo meiosis 2, and they will produce 4 cells. That's it. There is no cycle again. Do you think that again they will undergo the same process again? No. So that's why meiosis is not a cyclical process. Please understand. But mitosis divisions, yes, they are the cyclical. That's why please understand, technically the term cell cycle means we are talking about mitosis only. That is the reason why if you are observing the NCERT content, in general, cell cycle, cell division. What is the information they are talking? Mitosis only they are talking. Understood or not? So in this way, with respect to the cyclical concept, you are talking only the mitosis. You are never talking the meiosis. But anyhow, for the cell division, here cell is dividing into one cell dividing into 4. That's why this is a cell division. Mitosis is also cell division. Meiosis is also cell division. But cycle term is used only for mitosis. You should not use for the meiosis. This is one of the most important points that you need to remember because many students are making mistake here. So that's why this is the first point you need to keep in mind. So in the general information they are talking means they will talk with respect to mitosis only. See, in the case of meiosis, what happens? Gametes are produced. In the case of males, sperm cells. Animals, if you talk, males means gametes, sperm cells are produced. Females means ova are produced. If you're talking for the plants, males means pollen grains are produced. Females means egg cells will be produced. Yes or no? This is what the information is. But here these are for the ultimately the gametes. Gametes are useful, not useful for the same generation. Gametes are useful for formation of next generation. So that's why we are not talking about meiosis in general. In the general information, we are always talking with respect to mitosis only. This is the most important point. Many students are not understanding the significance of this. So please focus on this. So all the cells are reproduced by dividing into two. That means they are talking with respect to mitosis. And with each parental cell giving rise to two daughter cells. So that from the each parent cell, upon mitosis, how many are produced? Two daughter cells are produced. And each time they divide, whenever they are dividing, every cell will divide into two daughter cells. That's it. Next one. These newly formed daughter cells can themselves grow and divide. Again, the newly formed daughter cells will grow, and then again they will undergo the division, giving rise to a new cell population that is formed by growth and division of single parental cell and its progeny. So in this way what happens? So ultimately after some time what you can see? You can see a large progeny cells. From where they are produced? From the initial single cell and its progeny cells. Upon continuous division, now you can see a large population of the cells. That's what they are saying. In other words, such cycles of growth and division allow a single cell to form a structure consisting of millions of cells. Because of this cyclical continuous divisions, what happens? From the initial single cell, you can observe, you can identify a large organism having millions of cells. In this way, the single cell zygote is able to undergo continuous divisions to form a large individual having millions and billions of the cells. This is what the basic introduction they are saying. So in this way, cells has to undergo some division. They are having different roles. So to fulfill those, they have to undergo some divisions. And generally we are talking about the two types of divisions. One is mitosis, other one is the meiosis. But slowly we will go into those things. But since it is introductory, so we will talk only about the introduction. So in this way, I hope now you understood how single cell is able to form large individual. And what is the division we are mainly focusing? Mitotic division we are focusing. Clear? And now the next question is, why cells are dividing? Why they have to divide? This is what the logical important point. And actually this content of why cells are dividing, now I am discussing. But actually in the NCERT, where they have given this point, at the end of the mitosis, there is a heading called as significance of mitosis. Within the significance of mitosis, this information was given. So now I am taking that information and first we are studying that point, because this is the basic logical point that we need to understand. So that's why first we are studying that why cells divide. Why cells are dividing. Why they have to divide? What is the role here? What is the logical content here? So that we need to understand clearly. So here what I will do is, I will give the same information. Information is same only. But one part I will say as a general information. Suppose you are not a biology student. Then how I will tell you in simple words. So in that way, first I will explain you. And then more technically, we will study the same information. So now let us try to understand in the normal general information. Why cells are dividing? Let us try to understand this. Why cells are dividing? When the new cells are formed, the size of the cell is small. Yes or no? Newly produced daughter cells are small in size. Okay, cell size is small, fine. And what about the nucleus? Nucleus is also present in this. It is having so and so size. Fine. This is how it is present. And now the newly produced daughter cell, will it be present in the same size itself? No. So it is keep on growing. Cell is growing means increasing in the size, means volume is increasing. Now what happens? Slowly the cell is increasing in size. Slowly the cell is increasing in size. You have to observe this one. So the overall volume of the cell is increasing. Very good. When the cell volume, cell means what? Cytoplasm is keep on increasing. When the cytoplasm volume is increasing, then what about the nucleus? Is the nucleus increasing or not? Yes, that is also increasing, but nucleus size is increasing, but the increase in nucleus volume is less. Increase in nucleus volume is less when compared with cytoplasmic increase. This is the most important point. When the cells are growing, when the cells grow, what happens? They are increasing in size. When they are increasing in size, now what happens? cytoplasm content is more increased when compared with nucleoplasm increase. Both cytoplasm and nucleoplasm are increasing, but the increase in cytoplasm volume is much higher when compared with nucleoplasm volume. Now because of this there is a problem. What is the problem? If the cell is, suppose if cell is keep on increasing like this, what happens? Nucleus size is not increasing much, but cytoplasm is increasing quickly. Now there is a problem. What is the problem? After a particular point, nucleus may not be able to control all the activities. Please remember, nucleus is called as brain of the cell because nucleus is controlling all the cellular activities. If cell is keep on increasing, now what happens? All the cell is increasing means many reactions, many components, many things are needed. And how a small nucleus can control all this? So if cell is keep on increasing, after a particular point, the nucleus may not be able to control all the cellular activities. Then what happens? Nucleus will fail to control the activities. If nucleus is failing to control activities means that will become abnormal. Abnormal cells are dangerous. Please understand how they will react and what they will do, so that we cannot predict. So that's why the system has adopted a mechanism. What is the mechanism? So it will allow the growth up to a particular point. Once it is reaching a particular point, that means up to this it can control. If it is further increasing, then it will create a problem. So until then it will grow. And then what happens? Now this cell will divide into again smaller cells. Now this large cell will divide into two small cells again. Again those cells will keep on growing, and until when they are reaching a particular size, again they will divide and form the small cells. So in this way, cyclically this process is happening. So in this way, one cell will divide into 2, 2 will become 4, 4 will become 8, 8 will become 16, 16 will become 32, like this. They are keep on dividing and dividing. Understood or not? This is what the simple logic. Once again, let us go for the conclusion. What is the simple general information? When the cell is keep on increasing in size, after a particular point, nucleus may not be able to control all the activities. That's why after growing to a particular size, cell will divide to form again smaller cells. This is what. So that in the smaller cell what happens? The more control it is having. So that is the reason why again it is dividing into smaller cells. So this is the reason why it is dividing. This is the general simple information. Over. So this one is over. Now the same information you need to talk technically, more technically. How you will talk about the technical words? Now let us see this. Same diagram, let us see here. Small cell increasing in size. Now directly I am going for the bigger cell. Okay, like this. Now what about the nucleus volume? Nucleus is like this. Nucleus is increasing, but its growth is very less. It is like this. Okay, now let us see. Now here what we are doing is we are calculating nucleocytoplasmic ratio. We need to calculate, technically what we need to do, we need to calculate nucleocytoplasmic ratio. What is this nucleocytoplasmic ratio? Nucleo N, cytoplasmic C. That's why it is called as N by C. Nucleocytoplasmic ratio. Small n you are writing, small c you are writing. So please understand this nucleocytoplasmic ratio. What is this nucleocytoplasmic ratio? It is the nucleus volume. That means volume of nucleus divided by cytoplasm volume. Cytoplasm volume. That means volume of nucleus divided by volume of cytoplasm. This is what nucleocytoplasmic ratio. So which ratio you are taking? Actually volume ratios you are taking. It is the volume of nucleus divided by volume of cytoplasm. So here there is some volume of nucleus, you just take that value, and there is some volume of cytoplasm, take that value, and you will get some answer here. Suppose you are getting some x value here. Now what happens here and what happens here? Tell me with respect to x. In this particular case here, is the value more than x or less than x or equal to x? Here in this case, is it more than x or less than x or equal to x? Think logically. Think logically. What happens to nucleus volume? Here nucleus volume is increasing, but that increase is very small. But what happens to cytoplasmic volume? This increase is very high. So the cytoplasmic volume increase is very high. That means the denominator value is keep on increasing very high. If the denominator value is very high, then what happens? You will get different values there. And that too, if you are observing the relation, where you will get the maximum value, this value is more. That means nucleus volume to cytoplasmic ratio is more for x. X is greater than y, that is greater than z. Yes or no? Yes or no? Here z value is very less because cytoplasmic volume is, this value is increasing. If the denominator value increases, what happens? The total value will decrease. So that is the reason why what happens. Smaller cells will have larger nucleocytoplasmic ratio value. They will have larger value here. Whereas the larger size cells will have smaller value for this particular one. I hope you can understand this. This is what the reason is. This is what the logical and technical details that you need to understand. Clear? So now what happens? That means technically what you are saying, once it is growing to a particular size, now again it is dividing to get back again its original normal value. That means again it is becoming smaller size. That means what is the technical word? So as the cell increases, as the cell size increases, nucleocytoplasmic ratio decreases. Here it is high, here it is less. Yes or no? As the cell size increases, what happens to nucleocytoplasmic ratio? The nucleocytoplasmic ratio decreases. So once it is decreasing means it is increased greatly. Now it will divide to restore. To restore. To restore means what? To again get back its original value of normal, this one. So in this way, the large sized cells will divide. Why? To restore their nucleocytoplasmic ratio again. If the nucleocytoplasmic ratio value is high, now the control is very good. Control is very good. If the nucleocytoplasmic ratio is less, the nucleus control over the cell is very less. That is the reason why they are trying to get more value of nucleocytoplasmic ratio. That means the cells are growing in size and then they are dividing to restore nucleocytoplasmic ratio like the normal. That means they are dividing to form a smaller cell. This is what the technical information. General information, we are saying that as the cell is keep on increasing in size, nucleus may not be able to control all the activities. That's why it is dividing into smaller cells. This is the general information. What is the technical information? As the cell is increasing in size, nucleocytoplasmic ratio is decreasing. That's why it is dividing to restore its original normal nucleocytoplasmic ratio. This is what the technical information. That's it. This is the reason why they are dividing. Over. Clear? So with this, why they are dividing, that point also we have completed. Clear? Next one here, this is some general introductory paragraph that they have given in NCERT. So in this paragraph they are saying some information. What is a cycle? Cycle means it is the sequential events. That means this information we are talking with respect to mitosis. Please understand this cycle, the term cycle we are using with respect to mitosis only, not with respect to meiosis. So in the case of cell cycle, there are some phases. What are these phases? G1 phase, S phase, G2 phase and then M phase. Starting from G1 up to G2, already we studied about this one in nucleus. So G1 plus S plus G2, what this portion is called? Interphase. It is called interphase. In the entire interphase, what happens? Cell is keep on increasing in size. That's why as soon as M phase is completed, newly formed cell is having very small size. Now in the G1 phase, it is increasing cytoplasmic growth. Growth means cytoplasmic growth is a continuous process. It is growing in G1 phase, growing in S phase, even growing in the G2 phase. That means by the time it is completing the G2 phase, it is getting the maximum size. So now it is dividing into two small cells. So in this way, this is what happening. So different process, different things that are happening in the G1 phase. Cell size will increase. This is a summary. So I am simply saying the summary. From the next lecture onwards, slowly for individual steps we will study. Okay, more details. In the G1 phase, what happens? Anyhow, cytoplasmic growth is taking place, and the majority of the cell organelles, they will start undergoing the duplication, and then cell will getting ready for the remaining division process. Once it is crossing G1 phase, entered into S phase means in majority of the cases it will complete the cell division. That's why whether it should undergo division or not, that decision will be taken at the G1 phase. Once it is entering into S phase means it should complete the cell division. So all these decisions will be taken in the G1 phase. Understood or not? Next one. Once it is entering into S phase, in the S phase what happens? DNA replication occurs, chromosomal duplication occurs. So in this way what happens? If after completion of M phase 2 cells are formed, now here only single cell is present. If 2 cells are formed, for both the cells genetic material is required. So that genetic material will be divided in this S phase. So genetic material division will occur. Genetic material means DNA division will occur in S phase. So this DNA replicated or duplicated in S phase will be distributed into daughter cells at the end of mitosis, M phase, mitotic phase. Clear? That is what the significance of S phase. And then G2 phase, this is also once again the gap phase. So here also growth is keep on happening, and the maximum growth will occur in this G2 phase. Please understand this, maximum cytoplasmic growth occur in G2 phase. And once G2 phase is completed, enter phase is completed, and now it will enter into the M phase. M phase is the actual cell division phase. So there actually the cell division will occur. At the end of this, again the 2 cells are produced. So 2 cells produced means again they are present in the G1 phase. And if they want to divide, again they will go another round, another round, another round, another round. They will keep on dividing until there is no further requirement. Until then they will divide. So in this way they are forming a large population of the cells. So in this way some sequence of events are occurring, and they are repeated. That's why we are using this one as cell cycle. So these are the general information. Okay. So in that paragraph they have given some other points, but anyhow they are general information. Slowly we will study all these points. Okay. And at the end of the paragraph they have given one sentence. What is that sentence? These cell cycle steps. So it should enter from G1 to S. From the S it should enter into G2. From the G2 it should enter into M. In the M again different phases are there. So all these moving from one stage to other stage, all these are under the genetic control. What is the meaning of genetic control? So that means each and every step is controlled by the genes, So genes are producing proteins, and those proteins are controlling these activities. That's why please remember, cell cycle is highly, it is under the control of genes. That is the reason why, so in the class 12 there is a chapter called human health and diseases. In that health and diseases there is a subtopic called as cancer. So you will study about the cancer. So please remember, here what we are saying, here we are saying that all these steps are highly regulated by the genes. If the genes are failed to regulate this, now it may lead to that uncontrolled proliferation called tumors. They will lead to the formation of the tumors, right? So benign tumors, malignant tumors are called as cancers. So that's why please remember this. So there are some genes that are controlling cell cycle, cell division. So that's why it is very important. So that's why all these steps are under the control of the genes. That's why they are under genetic control. This is what the simple word they have given. But anyhow, in the health and diseases, so how these genes are controlling, so what are the genes, what happens, what is the problem, all these things we will study. But anyhow, even in this particular chapter also, some points related to that control regulation also I will tell you separately later on. Okay. With this introductory paragraph is also completed. Now let us quickly go through point by point and let us try to understand this. Some basic information only, nothing difficult here. Cell division is a very important process in all the living organisms. Yes, for all the living organisms, cell division is very important. Next one. During the division of a cell, DNA replication and cell growth takes place. So during the cell division, what happens? DNA replication, where in the S phase, DNA is also replicating and cell is also growing in size. Cell is growing in size, and inside the nucleus, DNA is undergoing replication. So DNA replication is occurring and cell growth is also taking place. Next one. All these process, that is cell division, DNA replication and cell growth. Here cell division means M phase, please understand. DNA replication, cell growth and cell division means M phase. All these things, so all these process hence have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genomes. So all these steps should be highly coordinated. Suppose one is happening before, one is happening later on. Suppose in a particular stage, whatever the process that have to happen, that should happen there only. Suppose they are delayed there. Suppose something that should happen in the next phase is happening now itself. That is also a problem. So that's why all these things should be highly coordinated, so that ultimately into the daughter cells intact genomes, that means without any damage, whatever the DNA that is present in the parents, the same type of DNA should go into the daughter cells. In this way, intact without any damage, it should go into the daughter cells. Next one, the sequence of events by which a cell duplicates its genome. So genome will be duplicated, DNA will be duplicated, means making a copy of DNA in the S phase, right? Next one, synthesizes the other constituents of the cell and eventually divides into two daughter cells is termed as cell cycle. So here you need to understand the word that is called as the sequence of events. So it is a complete sequence. There is a fixed sequence. You should not change that one. There is a fixed sequence. In the same sequence they are happening. So this sequence of events where different things are happening here, so that ultimately they are distributed into the daughter cells, and this sequence of events is called as cell cycle. Although cell growth means here that in terms of cytoplasmic increase. Some cases cell growth means one cell is dividing into 2 cells, 4 cells, 6, 8 cells. That is also called as growth with respect to number. But here we are talking with growth with respect to size. So what is this growth here you are talking? They are talking about cytoplasmic growth. Cytoplasm is increasing, right? That type of growth they are talking. Although cell growth in terms of cytoplasmic growth is a continuous process. That means in the G1, S phase, G2 phase, in all these phases, cytoplasm is keep on increasing. It is keep on growing. That's what they are saying. It is a continuous process. But DNA synthesis occurs only in one specific stage. What is that stage? In which stage the DNA replication occurs? Only one stage they have given, right? So underline this, during only one stage. What is that stage? S phase. Anyhow, slowly we will study from the next lecture onwards. So DNA replication occurs only in S phase, but cytoplasmic growth is happening in all the phases. All the phases, mainly in the interphase it is taking place, okay, of the cell cycle. Next one, the replicated chromosomes means DNA. When they are replicated in the S phase, so the replicated chromosomes are then distributed to daughter nuclei by a complex series of events during the cell division. So in the cell division, we will study a stage called as a telophase. In the telophase, what happens? Two nuclei are produced. So in the S phase, DNA is replicated. That replicated S phase DNA is now ultimately going into daughter nuclei in the telophase. After the telophase, cytokinesis will occur, so that now they are distributed into two new daughter cells. That's what they are saying. Clear? Next one. By a complex series of events, slowly we will study all these series of events during the cell division. These events are themselves under the genetic control. So all these stages, all these events are under the control of genetics. That means genes are controlling all these things. So genes will produce some proteins, and those proteins will control each and every step. Each and every step. Suppose a cell has to move from G1 to S, proteins will decide. From S phase to G2 phase, some other proteins will decide. So in this way, from every stage to next phase, so this movement is controlled by the genes. So genes will ultimately control all these things. That's why they are under the genetic control. If these genes are mutated, then what happens? The cell cycle control will be lost. If cell cycle control is lost, they will divide continuously and they may form some tumors, and that is dangerous for the cell. Okay, so this is what they are saying. This is what about the cycle they are saying. And now they are talking about the duration of the cell cycle. And this paragraph, I will read it. But before reading this, there is one important conclusion here also. You need to understand another important conclusion. What is that important conclusion? Observe carefully. When we studied about the cell chapter, there under the heading of prokaryotes, we studied one sentence. What is the sentence? Prokaryotes are generally smaller and multiply more rapidly than eukaryotes. This is what the word they said. Multiply more rapidly than eukaryotes we have studied. At that time, what I said? I said that for the E. coli, the division time is 20 minutes, and for the yeast, it is 90 minutes, and for the human cell, it is taking approximately 24 hours. This is the information we studied now. And from where we got this particular information? Yeast is dividing in 90 minutes and human cell is dividing in 20 minutes. Now itself. Now whatever the paragraph that we are going to study, that paragraph has this information. Then what about this particular one? E. coli will divide in 20 minutes. This information was given in class 12th NCERT molecular basis of inheritance. There is an experiment called Michelson and Stahl experiment. There they have given this value. Clear? Nice. But here the information is E. coli is a prokaryote, but yeast and human are eukaryotes. Yeast and human are eukaryotes. Very good. Now there is an important question. What is that question? Cell cycle cell division concept. Cell cycle cell division concept, where we are studying G1, S, G2, M phases, right? This type of cell cycle cell division concept is applicable to only prokaryotes, only eukaryotes, both. Tell me the answer. Which one is the correct one? So the cell cycle concept having G1 phase, S phase, G2 phase, M phase, likewise these phases are there, no? And these phases containing cell cycle concept is applicable to only prokaryotes, only eukaryotes, or both. This is the most important point. Many students are not understanding this significance. So this is applicable only for eukaryotes. Please understand, only for eukaryotes. So these phases of cell cycle like this, you are studying only for eukaryotes. So the cell cycle cell division concept, whatever we are studying, this concept is applicable only for eukaryotes. It is not applicable to prokaryotes. That means if I am asking a question about the E. coli, in the E. coli you cannot say G1 phase, S phase, G2 phase, M phase, because E. coli is a prokaryote bacterium. In the bacteria these phases are not present. These phases are present only in eukaryotes. These are never present in the prokaryotes. That's why, what is the conclusion? That's why in this chapter cell cycle cell division, you just go through the entire chapter. Nowhere they are talking about the prokaryotes, because this concept is not applicable to prokaryotes. The mechanism of division, cell division, or the fission, or the different mechanisms are there. They are different in the case of prokaryotes. They will not follow this particular mechanism. So this is the most important point. So in this cell cycle cell division chapter, we are not talking about any prokaryote. We are talking only about eukaryotes, because this concept is applicable only for eukaryotes. This is the one important point. So that's why you are not studying about prokaryotes here. We are studying only about the eukaryotes. Good. Now in this way, now you just say this is also, yeast is also eukaryote, human is also eukaryote. Now based on this also you can say this one. Do all eukaryotes have same time for the cell division? That means suppose it is starting from G1 and it is going through all these and again coming to this one. That means this is the one complete cycle. For one complete cycle in the yeast, what is the time it is taking? Just 90 minutes. Just 90 minutes, right? So 90 minutes means what? So 1 hour 30 minutes. That means 1.5 hours. That's it. But here it is taking 24 hours. Now based on this, what is the conclusion? All the eukaryotes do not have same generation time. It is called generation time. Is the generation time same for all the eukaryotes? No. So different organisms will have different duration of cell cycle. Different organisms will have different duration of the cell cycle. This is what the one important point that you need to understand. So with respect to different organisms, you are saying. Very good. Now we will focus only on one individual. Let us talk only about the human. Now in the human again, there are different types of cells are present. At different locations, different cells are present. Now the question is, only for the human I am asking, for the human only. Do all the cells present in single individual have the same time for division? Question is very technical, observe carefully. Do all the cells present in one individual have exactly same duration? It is not always true. Please understand this. They are not always having the same time. If you take this human skin cell, Generally, this 24 hours whatever we are talking, this is generally for the skin cell division. For the normal skin cell division, that too in the culture laboratory we are studying this one, okay. So in the laboratory conditions, so skin cells of human, they are taking approximately 24 hours for the division. This is for the skin cell you are talking. Now you just imagine the stomach. In the stomach, what happens? There is a epithelial lining is present. There also some cells are present. Now, do you think that those are also dividing for 24 hours? No. Please remember, in the stomach, HCl is produced. When HCl is produced, when it is acting on the food, it will also damage some of the cells of the stomach. Some of the cells of the stomach. So that is the reason why those cells are quickly dividing, very quickly dividing. So that is the reason why even in single individual also different timings are possible depending on the location of the cell. Now again different generation times are present. So that's why we are taking one common example for our study purpose. So here we are talking about the human cell. Generally it is the skin cell that is having 24 hours duration for completion of one cycle. For one cycle it is taken 24 hours. Now what they have done in NCERT is, in the next coming explanation, they have used this timing, and as per this they have given the remaining timings, okay. So we will also go according to that particular information. Clear? This is what one important point that you need to understand very clearly. So this concept of cell cycle phases is not applicable to prokaryotes, it is applicable only for eukaryotes. Even in the eukaryotes also, different organisms are having different cell cycle times, cell division times, and that too even in single individual also at different locations different timings may be possible. That's what the conclusion is. Now let us go through this information. Duration of the cell cycle. A typical eukaryotic cell cycle is illustrated by human cells in the culture. So human skin cells in the culture conditions, we are taking them for our study. And these cells divide once in approximately every 24 hours. That means for every 24 hours they are dividing. Okay, this is what they are saying. Next one. However, this duration of cell cycle can vary from organism to organism. So in different organisms, so that cell division cycle time is, cell cycle division time is variable. And also from cell type to cell type. What is the meaning of this cell type to cell type? Even in the single individual also, different cells may have different timings. This is what the meaning is. And yeast, for example, can progress through the cell cycle in only about 90 minutes. So human skin cells are having approximately 24 hours, but yeast cells can divide only within 90 minutes. So in this way, different cells are having different generation times. This is what the information that they have given. And after this is the most important content that you need to understand. So now I am going through this. You need to compulsorily know about this. If you are not understanding this, again you will face a huge problem while you are going with the phases. In the phases, general information is okay, but many times they will ask with respect to what is the ploidy in so and so stage and what is the DNA amount or DNA content in so and so stage, they will ask. So this information also you need to understand. So now I will try to explain this in simple, but anyhow in the cell chapter we studied about the ploidy content, so you can easily understand this. Okay, so please remember, already I told you what is chromatin, what is chromosome. If it is present thread like chromatin, if it is condensed rod like chromosome, that is the difference, right? Now we are talking here, and for your understanding purpose, I am giving entire details with respect to human. I am giving entire details with respect to human so that you can easily understand. Okay, the same logic you can apply to any organism, but for human you can easily understand. That's what my aim is. So that's why now let us see here. Now what I will do is I will give some conditions. I will give some conditions, and you need to observe the conditions, and then you need to tell me what is the ploidy. Whether it is n. N means what? Haploid. If it is 2n, that is called diploid. If it is 4n, that is called tetraploid. Please observe these ploidies: haploid, diploid, and then tetraploid. Okay, which one is exactly? Based on this condition you need to tell me, and at the same time you need to tell me DNA content or DNA amount. DNA amount is indicated with capital C. Capital C, I will tell you what is capital C. So based on this you can write whether it is capital C or 2C or 4C. What is it exactly? That you need to write. That's it. These are the two things that you need to understand. With this lecture will be completed. So please observe carefully. First, let me give you some conditions. What are these conditions? Observe carefully. Suppose it is a single thread-like structure, or for the same thread, let us make it into a condensed form. Suppose it is a single, here also it is condensed chromosome and having only one chromatid. Observe this. Suppose like this, there are 23 in a cell. Suppose like this, there are 23 in a cell. Then what is the ploidy? What is the C value? That you need to observe. Second one is, if they are single thread-like structures or condensed chromosome having one chromatid. Suppose like this, if 46 are present in a cell, then what is the ploidy? Then what is the DNA amount? Third one. This time we are drawing about double threads. Double like this, but both of them are attached at a common centromere. Observe the diagram carefully. Two thread-like structures are present, but both of them are attached at common centromere. Like this, 46, 23 are present, or like this, 23 are present. Please observe like this. What is the meaning of this? Single thread like 23, or condensed rod like single chromatid ones, they are also 23. Like this 23 or like this 23, whatever it is. What is the answer you just write? Like this 46 or like this 46, whatever it is, write the answer. Like this 23 or like this 23, write the ploidy and DNA content. And the last one is now like this, or like this, 46 are present. Now tell me, what is the ploidy, what is the DNA content? This is the most important concept. Many students are making mistake here. If you don't know this basic information, definitely you will make mistake when we are studying about prophase, metaphase, anaphase, telophase, all these things. Okay, there also, and even in the G1 phase, S phase, G2 phase also. If you don't know how exactly they are present, so then again you will make a mistake. So like this only, very clearly I will represent every information in the regular lectures from the next lecture onwards. Now tell me quickly, let us complete this fastly. Tell me, single threads like this or single condensed ones? If 23 are present, what is the ploidy with respect to human? So what is the condition here? Like this 23 or like this 23, whatever it is. 23 are present. 23 means it is one set. 23 means it is one set. That means it is haploid. It is haploid. Observe carefully. Okay. Now I will give you another condition also. At some location you will get like this. Like this also you will get. So that's why I am writing this one. Okay. So now let us see. So in this 23, if they are present, it is haploid condition. First we will fill the ploidy. That is haploid here. Like this or like this. If 46 are present. So please remember, whenever, see here, the ploidy we are giving based on the chromosome number. Please understand this concept. You need to understand. Ploidy you are giving based on number of chromosomes. If single set having 23 chromosomes, that is haploid. Two sets 46 chromosomes, that will be diploid. Likewise, to say what is the ploidy, you need to focus on how many chromosomes are present. Yes or no? And how you will calculate the chromosomes? There is a blind information is there. Please observe carefully. One centromere is equal to one chromosome. Please remember, that's why this is one chromosome, this is also one chromosome. Why this is also one chromosome? One centromere only is there. Don't worry about whether it is having one chromatid or two chromatid. This diagram is having two chromatids, but this diagram is having one chromatid. We are not worried about chromatids. Our focus is only centromere. So in this diagram here, how many centromere? Only one centromere. Like this, how many? 23 are there. So 1 centromere into 23, that means total how many? 23 centromeres are there. 23 centromeres means how many chromosomes? 23 chromosomes only. If 23 chromosomes are there, what is the ploidy? Haploid only. So whether thread-like or condensed, it will not make any difference. It is the same only. So in this way, 23 chromosomes, that's why haploid. Now 1 centromere, anything you can take. 1 centromere, like this 46. So 1 into 46, 46, that means it is diploid. It is diploid. Understood the logic? Next one. Here what happens? Anywhere you just take only one centromere. Likewise, how many are there? 23. Don't worry about two chromatids. Don't see chromatids. You just see centromeres. One centromere into 23. That means 23 centromeres. So 23 centromeres means 23 chromosomes. 23 chromosomes. This will be haploid. That's it. Next one. Here one centromere. Likewise 46. That means 46 chromosomes are present. If 46 chromosomes are present, it is diploid. Understood or not? Next one here. One centromere into 92. That means 92 chromosomes. 92 chromosomes means it is a tetraploid. It is a tetraploid. It is tetraploid. This is what the information. This is how in different stages you will observe the chromosomes or the chromatin like this. So you need to know. So simply by looking at the diagram, you need to know what is the ploidy. That is the reason why in different phases they will ask that. In the metaphase of mitosis, what is the ploidy? In the anaphase 1 of meiosis, what is the ploidy? They can ask any question. So first thing you need to know is whether it is thread-like or condenser rod-like. If one of this one is there, then single threads are there, double threads are there, that you need to observe. And then like this, how many are present? Once you know this information, that's it. In the entire mitosis meiosis, only these 5 only you will repeat. These 5 only will be repeated at different locations. That's why we are studying this one first. Completed. Next one. Next we are going with the C value. What is C value? Please observe here very carefully. In the human haploid, how many chromosomes are present? Let us take the sperm cell or the ovum. With respect to human, I am saying, right? In the human sperm cell or the human ovum, how many chromosomes are present? 23 chromosomes are present. How those 23 are present? Like this. Like this 23. 23 are not like this. In the sperm cell or the ovum, how they are present? Like this, number one condition. Like this 23 are present. So now you need to calculate the DNA molecules. What is this one? DNA amount or DNA content. So you need to count DNA. So how many DNA are present? See this single thread, inside this single thread, one double stranded DNA is present. If it is condenser rod like this, one chromatid, only one double stranded DNA is present. So whether it is like this or like this, one double stranded DNA is present. One double stranded DNA present in 1 into 23. That means 1 into 23. How many? 23 DNA molecules. Here 23 DNA molecules. Understood or not? Now let us see here. 46 into each one is having 1. So 1 into 46. That means here 46 DNAs. Here 46 DNAs are present. First I am writing the DNAs. Based on that, whether I will write you whether it is C or capped. Single C, 2C, 3C, 4C. What it is, that we will write. Now here 23 into. Now observe carefully. Each thread is having one double stranded DNA. That means here how many threads? 2 threads are there. That means how many DNAs? 2 double stranded DNAs are there. 2 double stranded DNAs are there. So 2 into 23. 2 into 23 is what? 46 DNAs. 46 DNAs are present. Here also, condensed also, same only, right? 2 DNAs into 23, same 46. Now here what happens? 2 DNAs into 46. 2 into 46, 92 DNAs. 92 DNAs are present. Understood or not? Next one. Here, this is one chromatid. One chromatid will have one double strand DNA. One double strand DNA into 92. 1 into 92, 92 DNAs. 92 DNAs. Like this. First we are writing how many DNA molecules. Now you can easily understand now. In the normal gamete cells. What is the normal gamete cell? Sperm cell or the ovum. That means here this is the condition. In the normal sperm cell or the gamete cell, whatever is the DNA that is present, that DNA amount means what? You need to measure. So here what happens, the amount of DNA is very small, so you cannot say in the grams. You are weighing it, but you cannot say in the grams. Grams is a very big value. So here what we are saying, in the picograms you are saying. What is the amount of this one? In the human cells, it is approximately 3 picograms. If you are weighing it for this haploid cell, what you will get? Approximately 3 picograms. 3 pico means what? 10 to the power minus 12 grams. That much is the weight. And that 23 DNAs, the DNA, amount of DNA of this 23 is called as 1C. 1 capital C. the DNA content, amount or content. Please understand, indicated with C for content, okay? Indicated with 1C. What is 1C? 1C is the amount of DNA present in one gamete. One gamete, it may be one sperm cell or one ovum. Approximately, it is approximately equal to 3 picograms. That is the 1C. So, for 23 DNA molecules, it is 1C. Then for 46 DNAs, what it will be? It will be double. So, it will be 2C. Here also 46 DNAs. So, it will be 2C. But here it is 92 DNAs. For 23, if it is 1C, for 92, how much it will be? 4C. 4 times it is. Yes or no? 23 into 4 is 92. So, that's why it is 4C. And here also 92. So, it is 4C. I hope now you understood what exactly I am saying. So, how to calculate this ploidy? Ploidy is simply based on this number of chromosomes. We are not worried whether it is having one chromatid or two chromatids. We are not worried. So, simply based on the number of chromosomes, we are writing the ploidy. But for DNA content, what you need to do? Number of chromosomes also you need. And how many chromatids? Is it one only, two only? That also you need, because you need to multiply these two. So, then only you can write the total DNA molecules. Based on the total DNA molecules, you are writing the C value. So, that's why this is how you are writing the table. I hope now you understood this. If you are still making any mistakes, so please go through this particular part once again. Observe the content carefully, because I cannot repeat this every time. It will take so much of time. Please understand, okay? So, this is how you are writing this. That's it. In this way, now what we will do from the next lecture onwards, for every phase we will draw one diagram. In the diagram, I will draw whether they are present like this or like this. I will mention clearly. And how many are present, that also I will mention. That means one of this representation I will give you. So, simply by looking at it, you should write ploidy, you should write DNA content. That is your duty. So, that's why first practice all these things. You should know in this lecture itself, because from the next lecture itself, we will go with the regular content. Clear? And now we are going with the final information, that is, what type of chromosomes are these? Are they monad chromosomes or dyad chromosomes? Monad or dyad chromosomes. Here we are not making any difference between chromatin and chromosome. Both of them are considered as same only, okay? Because the same is converted into chromosome, same is converted into chromatin. Clear? Now tell me, what is monad? Mono, single. Single, that means chromosome with one chromatid. Chromosome with one chromatid is monad chromosome. What is dyad? One chromosome having two chromatids. Monad is one chromosome having one chromatid is a monad, and whereas dyad is one chromosome having two chromatids. Now observe the diagram. Here how many? Here one chromosome has only one chromatid. You can take this or take this. Anything you can take. One chromosome has only one chromatid. That means in this diagram, the type of chromosomes are monad chromosomes. Please remember this one. Once we are completing the entire chapter, one separate lecture I will spend for entire mitosis meiosis. There you need to write this entire particular table. So that's why I am giving entire information now itself, so that you can slowly practice all these things. Now one chromosome having, how you can say one chromosome, one centromere, that's why one chromosome. So one chromosome with one chromatid, that's why monad. Here also one chromosome with one chromatid, that's why this is also monad. Next one, but here what happens? One chromosome has two chromatids. Here also one chromosome has two chromatids. So that's why it is dyad chromosome. One chromosome with two chromatids is the dyad. Next one, here also one chromosome with two chromatids. That's why it is also dyad chromosome. What is the last one? One chromosome with one chromatid. That's why it is a monad chromosome. This is how we are writing this one. Clear? With this information is completed, and separately in the meiosis you will study about tetrad. Tetrad. What is this tetrad? Tetra means four chromatids we want. Can single chromosome has four chromatids? No. This is the chromosome having two chromatids. This side is one, this side is other one. Then how you will see this tetrad? That's why tetrad is not a chromosome. Tetrad is a condition. Please observe this one. Tetrad is not a chromosome. There is no tetrad chromosome. Tetrad condition is seen. So what happens in the tetrad condition? Two homologous chromosomes are attached. So here if you see overall two chromosomes are there. But in this condition, not chromosome, condition 1, 2, 3, 4. Total of 4 chromatids are present. That's why it is called a tetrad condition. It is called a tetrad condition. And this tetrad condition. So this tetra means 4. This tetra add is with respect to chromatids. Chromatids. So with respect to chromatids, if you talk, 4 chromatids are there overall. That's why it is called a tetrad condition. But the same is also called as bivalent condition. Bivalent condition. What it is bivalent? Bi means 2. What is 2? This is one chromosome, one chromosome. This valent is with respect to chromosome. So you need to understand technical words or else you will make the mistake. So overall 4 chromatids are present. That's why it is called a tetrad. But overall 2 chromosomes are present. So bivalent. So if 2 chromosomes are present together, that is bivalent. If chromosomes are present individually means in all these diagrams they are present individually only. Okay, please remember this is entirely one chromosome only. So one chromosome present individually. So all these are present individually. So all these are monovalents. Monovalent means single chromosomes. They are single chromosomes, separate chromosomes. They are not attached. But these are attached only in the meiosis, that too only few stages. Majority cases you are observing monovalent only. Understood or not? So totally you have studied 4 types of names. What are these 4 types of names? First one is with respect to ploidy you have observed. Second one is DNA amount or DNA content you studied. And then you studied about whether it is a monad, dyad or tetrad condition. Okay, that also you studied. And then you studied about whether monovalent or bivalent. So this is the information that you have studied in this particular lecture. So with this lecture is completed, but this last content is very important, very important. Please practice very carefully. You should not make any mistakes here. Clear? With this particular part is completed. So from the next lecture onwards, we are going with the regular mitosis phases one by one we will clearly study. Okay. Thank you so much, my dear friends. We will meet in the next lecture. Thank you.